Unit 17, Human Humanity by Faye Redshaw. So what is an infection? An infection occurs when a microorganism, which is not usually present within the human body, enters a person's body. This typically occurs through the nose, mouth or cuts in the skin and causes harm. The microorganism uses the body to sustain itself, reproduce and colonise. These organisms are known as pathogens. Not all infections show symptoms, which is why some can be hard to identify. So types of infection, there's 4 types of infection. You've got bacterial. This is a single-celled organism responsible for illnesses such as UTIs, strep throat and pneumonia. Viral, these rely on the human body as a host to reproduce as they are not alive themselves. They are responsible for many diseases ranging in severity from common colds to AIDS. Fungal, this is responsible for many skin disease infections. They live in the dirt, plants or household surfaces. These infections can cause problems such as bumps or rashes and problems to the lungs or nervous system. Examples include athlete's foot and ringworm. And parasites, these are organisms that live on or in a host organism and get their food from or at the expense of its host. Malaria is a serious fatal disease caused by a parasite which is transmitted by a mosquito bite. Other parasites can be transmitted to humans from animal feces. So the main types of disease transmission are direct contact, one of the easiest ways to catch a disease by person to person, animal to person or mother to unborn child. Indirect contact, when somebody may cough or sneeze onto an object, leaving their germs behind for the next person to touch. Infections can last up to several days on an innate object. Insect bites, these pass on diseases such as a mosquito bite, which can cause malaria, and food contamination is a disease causing germs may infect a healthy person through contaminated food or water such as salmonella or E. coli. So there's 3 lines of defence. Your immune system is made up of 3 lines. The first 2 lines of defence are non-specific mechanisms that innate from birth, unlike the 3rd line of defence, which is a specific response. Non-specific defence mechanisms respond to all pathogens in the same way. They are effective against a wide range of pathogens and stop them entering the body. If a pathogen passes through one level, the next level will take over. So the first line of defence, this consists of physical and chemical barriers to prevent entry of the pathogens to the body. The skin is the largest organ of the body and acts as a barrier to infection. The skin prevents penetration of chemical toxins and microbial organisms. The skin forms part of the integumentary system and comprises of 3 major layers. So the epidermis, this is the outermost layer. It has a waterproof protective coating to protect from external environment. It replaces itself every 28 days with new skin cells. It contains melanin, which protects against the sun's UV rays. The dermis, which is underneath the epidermis, it supports the epidermis and it enables the skin to thrive. It also plays a number of other roles due to the presence of nerve endings, sweating glands and blood vessels. And the subcutaneous, this layer forms the deepest section. It acts as a layer of insulation due to the fat being stored here. It also protects internal organs and muscles. So other chemical and physical barriers. So you've got the mucous membrane. This is the primary interface between external and internal environment, which lines surface such as eyes, nose, mouth, ears and throat. They are coated with secretions which fight microorganisms and create a defence to infections such as tears and saliva. The gastrointestinal system, so the acidic and alkaline secretions act as a protective system against infections. Intestinal contractions help remove unwanted microorganisms. The female genital tract, the acidity of the vagina prevents harmful bacteria from growing. The urinary tract, this is made up of numerous barriers to infection. The bladder is protected by the urethra, which is long enough to stop bacteria passing through it. Frequent urine helps remove pathogens. However, breach of these first line barriers, such as getting a cut, would allow bacteria to enter the body. This is when the second line of defence would respond and take over. So the second line of defence, is a group of cells, tissues and organs working together to help protect the body. These are non-specific cellular and molecular responses of the innate immune system. They respond to every pathogen in the same way. Phagocytes recognise when something enters the body that should not be there. Phagocytic leukocytes are intrinsically involved in the inflammatory action. They travel to infected areas and destroy the items they wish to remove. Blood circulation increases around the infected area, causing inflammation. This causes swelling around the localized area. Fever increases body temperature, which activates heat shock proteins, which stimulate the production of pro-inflammatory cytokines and stops bacterial growth. And generally, the second line defence is enough to fight off the infection. However, sometimes the innate system cannot control the infected area. This may be due to much bacteria, and the body has to rely on the adaptive immune response to intervene. So, acquired immunity, the third line of defence. Acquired immunity is not present at birth, it is learnt through exposure to the environments in which we live. The body's immune system learns how to attack each antigen which enters the body. This learnt defence consists of cells which eliminate specific microorganisms that have invaded the body's tissues. Acquired immunity can be developed actively or passively, with both being either naturally or artificially developed. So, leukocytes. These are white blood cells. These help defend the body against infections. They are produced within the bone marrow and live within the lymph system. They circulate around within the blood and are present in tissues throughout the body. The body consists of 5 different types of leukocytes that each play their own role removing unwanted antigens. So the neutrophils, these target bacteria and fungus. They are the first cell to travel to the site of infection. They help fight infection by ingesting microorganisms and releasing enzymes that kill the microorganisms. They make up to half of the white blood cell population. Eosinophils, these attack and kill parasites and bacteria. They also help with allergic responses. Basophils, these are responsible for fighting bacterial infections, viruses and fungi. And lymphocytes, there's two main types, the B cells and the T cells. So the B cells produce antibodies that are used to attack invading bacteria, viruses and toxins. And C cells, these destroy the body's own cells that have themselves been taken over by viruses or become cancerous. And monocytes, these clean up dead cells within the tissues and help production of cytokine molecules. So immunoglobulins. Immunoglobulins are proteins produced by plasma cells and lymphocytes which bind to antigens. They have many uses within the body. The first to appear when a pathogen is detected. They attach to foreign substances and assist in destroying them. Opsonization, a pathogen is marked for destruction. Neutralization, prevent it from infecting cells. Activation of complement, introduces system to destroy antigens. And agglutination, a clump particles together, so phagocytosis can occur more frequently. Activation of mast cells. So the types of immunoglobulins. So immunoglobulin J, this is the most common. It forms a direct attack to protect against bacteria and viral infection. The only immunoglobulin that is transferred from a mother to unborn baby to form some disease protection via the umbilical cord. Immunoglobulin A, this is the second most abundant. This provides cell defense for mucosal surfaces. Immunoglobulin M, this structure is different than the others. It's found mainly in the blood and lymph fluid. This is the first antibody the body makes when it fights a new infection. Immunoglobulin A, these levels increase significantly with parasite infections and allergy reactions. And immunoglobulin D, this is an uncommon molecule. It's part of the B cell receptor and activates with basophils and mast cells. So self-recognition and tolerance. To be able to protect the body from invaders, the immune system must recognise that it's self and non-self, distinguishing the difference between self-produced antigens which also responding to unwanted foreign substances. For the body to remain healthy and functioning effectively, it is important that there is the balance between immunological defence and self-tolerance. This is achieved through the mechanisms of antigens. The two most important factors which distinguish between self and non-self are the major histocompatibility complexes 1 and 2, as well as human leukocyte antigen gene complex. The function of MHC molecules is to bind peptide fragments derived from pathogens and display them on the cell surface for recognition by the appropriate T cells. Generally, human cells display specific types of antigens which the immune system can recognise as self. It therefore knows not to attack these cells. Alternatively, an unknown antigen will be recognised as non-self by the innate immune system and the T and B cells will respond. A stronger immune response will be activated for those antigens which are very different to that of the human antigens compared to the ones which are very similar. So MHC1 molecules are all nucleated cells used as an early warning up to the immune system. They degraded if foreign proteins are witnessed for the immune system T cells to find. MHC2, they work similar to MHC1, but only on the surface of antigen presenting cells. Antigen presentation with MHC2 is essential for the activation of T cells. So self-tolerance failure. So hypersensitivity. This is an overreaction of the immune system to repeated exposure to an antigen which triggers an immune response. According to the Jellcomb's classification of hypersensitivity, there are 4 classes, each varying in severity, from allergies as a result of pollen to multiple sclerosis. Autoimmunity, the immune system attacks its own body believing that there are foreign elements. This can destroy cell and damage organs. So rejection of transplanted organs. Transplantation of organs is transferring cells, tissues or organs via a donor to another person. This is to replace any damaged organs. However, the immune system's mechanism to fight any foreign objects can also reject transplanted organs due to the antigens of the cells being non-self. Therefore, closely matched antigens reduce rejection probability. There are 3 types of rejection. So hyperacute, this occurs a few minutes after the transplant. The transplant is completely unmatched. Acute, this occurs within the first few weeks up to 3 months after the transplant. And chronic, this occurs over many years due to the constant immune response slowly damaging the organ. So autoimmune disorders, the immune system helps keep your body healthy and protects against infection and disease. However, autoimmune diseases result in the immune system attacking healthy cells within the body. There are over 100 autoimmune disorders which can affect every part of your body, ranging in severity and treated differently. So multiple sclerosis, MS, this involves the protecting coating of nerve cells in the spinal cord and brain are attacked and damaged by the immune system. Damage to the myelin disrupts the signals being sent from your brain to your body, resulting in physical and mental problems. With degeneration increasing over time, symptoms include muscle weakness, balance issues and numbness. So diabetes, type 1 diabetes, the hormone insulin producing cells in the pancreas are attacked by the immune system. This affects the amount of glucose within the bloodstream, as insulin helps stop hypoglycemia occurring. Hypoglycemia may damage blood vessels and organs such as the heart, kidneys, eyes and nerves. Insulin injections are used to treat type 1 diabetes. So acquired immunity, so there's 4 types. There's 2 active and 2 passive. So active natural immunity, when the body is exposed to a pathogen and becomes infected with the disease or virus, the body's primary immune response fights the disease and attacks the pathogen. The T cells in the bloodstream attach to the pathogen and present it to the B cells in the bloodstream. Antibodies are produced by the B cells to kill the pathogen. As a result, the person becomes immune to the pathogen as the B cells remember the antibody if encountered again. And then active artificial immunity, when the body is initially exposed to an antigen via a vaccine to generate an active immune response. The immune system is stimulated to produce antibodies by the antigens in the vaccine. Memory cells are produced to fight future antigens of the same infectious agent. If future exposure was to occur again, the memory cells would be able to quickly attack the infectious agent before symptoms can develop. And then passive natural immunity. This is when antibodies are transferred from the mother to child. Prenatally, the mother transfers antibodies through the umbilical cord through her blood. This will protect the child up to the first 6 months. Postnatally, antibodies can be transferred via breast milk. Then passive artificial immunity, when the body receives an injection or a transfusion, when the antibodies have been made up somewhere else. However, antibodies gradually break down and therefore protection only lasts for a couple of weeks. The person's own immune system does not become involved in protecting against the infectious agent. So, immune response to vaccinations. As viral disease cannot be cured with antibiotics, vaccinations are needed to help protect against severe diseases. Vaccinations are implemented to help the body gain immunity against specific pathogens. They expose the body to a weakened version of that pathogen, stimulating the body's white blood cells to produce antibodies. A weakened version reduces the possibility of the person developing the specific disease, although side effects are experienced. Vaccines are weakened by containing harmless fragments of a pathogen. Live pathogens specifically treated to make them harmless. Toxins produced by pathogens or dead pathogens. Live vaccines are made through virus or bacteria that cause diseases, but they are weakened in a laboratory. Small doses are implemented into the vaccine, which replicates inside the body to create a stimulus response from the immune system. As they are small doses and weakened versions of disease, adverse reactions are lower and less severe than the natural disease. The immune system does not differentiate between a weakened version of a natural version. Therefore, the immune response is the same. A live vaccine imitates a natural infection, which stimulates a broader immune response. One dose is generally effective to create an immune response. Inactivated vaccines are produced through growing bacteria or viruses, then inactivating it with heat or chemicals. As they are inactivated, they cannot replicate, so pose no danger to disease from the vaccine. Although they require more than one dose, the first initial dose does not form a protective immune response. It prepares the immune system for future doses. It is with the second and third dose in which a protective immune response develops. So the benefits of the vaccinations. Vaccinations for measles began in the UK in 1968 and later with the introduction of the MMR vaccine in 1988. Since 1940, the number of cases and deaths has significantly declined. In 1940, there was 857 deaths caused by measles, alongside 409,521 cases. Since 1992, there has only been 2 deaths caused by acute measles. Although healthcare and sanitation levels have improved over the past 50 years since the vaccine introduction, there are significant benefits for the use of vaccines against disease, in particular measles. So herd immunity. This is the spread of contagious disease is reduced when a high percentage of the population is vaccinated due to the reduced numbers of people who become infected. Contagious disease spread directly from one person to the next. Therefore, it is a significant proportion of a community is vaccinated, the disease will struggle to spread any further. This is known as herd immunity. Once enough people are vaccinated, vulnerable people such as the elderly or newborns are helped in being protected, alongside those who cannot be vaccinated, such as children. Vaccination programs. The NHS vaccination programme are available to everyone living within the UK from 8 weeks old, with the first immunisations through to adulthood with seasonal flu vaccines. Vaccination programs are made to be easy and accessible to everyone. Pharmacists can now administrate certain vaccines to try to increase uptake numbers. Obviously, more frequently, the COVID-19 vaccination has been more available to all adults throughout the UK. So the factors of affecting vaccine uptake. There are numerous factors affecting the uptake, like seasonal variability. The prevalence of many infectious diseases can vary. Therefore, vaccination programs are rolled out seasonally in an attempt to control the spread of the disease, such as the flu, norovirus and bronchiolitis, as they can be very serious for specific groups, such as the elderly or young. Then you've got the anti-vaxxers, people who don't believe that vaccines do more. People believe that vaccines do more harm than good due to the have reduced uptake figures. And then you've got the conspiracy theories linking the MMR vaccine to autism. There has been a decline in the uptake of children having the vaccine. This has had a significant increase in the number of measles cases in previous years across Europe. And that's the end of my presentation. Thank you very much.